Aloha. How you doing? We left off here talking about birds. There we go, bird wings. And this was the discussion of structure and function. And I promise you to show you a picture of my pumpkins. Sounds exciting, doesn't it? So there's my pumpkin patch. It's a lot bigger than that, but that's a pretty good sized pumpkin right there. Picture doesn't do it justice. That's a gallon jug of water. And the pumpkin is much bigger than that gallon jug of water. This thing's huge. But you can see, this leaf is being eaten by the insects. How flat the leaves are, like a solar panel, because their job is to collect solar energy. So there's that. Moving right along. The cell is the basic unit of life. And by that, I mean that it's the smallest structure that can perform all the functions we associate with being alive. One of those, being able to reproduce, make a copy of the genetic material and divide into two. Another one, the use of having a metabolism, using a source of energy to maintain organization. So the cell is the basic unit of life. You get that? Good. The cells, all cells, now notice how that says all cells, not some cells, not most, but not all cells, but it says there, all cells. So this is a pretty big, broad biological statement. All cells have a membrane. We'll talk about the makeup of that membrane later. It's called the plasma membrane, and it's the boundary between the inside of the cell and the outside of the cell, kind of like the walls and doors of your house or where you live. All cells use DNA as to contain their genetic information. And there's a couple more. All cells use the same 20 amino acids to produce our proteins. And we have a whole section on proteins coming up soon. We have all cells use a molecule called ATP. Maybe you've never heard of ATP, but ATP is the energy, it's a molecule that delivers energy on site where it needs to, kind of like cash. Remember what cash was? You go buy something, you hand some money over. So it's kind of like cash, which is now more like our debit card. So there's four things that all cells have in common. Two of them are here, enclosed by the plasma membrane as a boundary between the inside and the outside, use DNA to store the genetic material, use the same 20 amino acid building blocks to make our proteins, and use ATP as an energy molecule. We have two types of cells in our planet, eukaryotic and prokaryotic. Pro is a term that means early or first, and karyotic is derived from the Greek word karyon, which means kernel or seed, like corn kernel is a seed. And so that refers to the DNA. And so prokaryotes are the earlier first way of packaging the DNA. And really, they don't have a package. It's just floating around inside the cell. The eukaryote means second or next or latest, is the newer version. And prokaryotes are much smaller. They're simpler. They're not simple. If they were so simple, let's just go ahead and make one. We can't do that. So even the simplest cell is very complex. Prokaryotes are much smaller than eukaryotes. They don't have their DNA confined inside the cell. It's just in a little region. And the only membrane, we'll talk about the membrane. I'll just go ahead and throw out the word right now, phospholipid bilayer. That means it's two layers of a molecule called phospholipid. And we'll take a much closer look at that later. So the phospholipid bilayer is what is meant by a membrane here. The prokaryote's only membrane is the plasma membrane, the outer membrane, whereas a eukaryotic cell has a plasma membrane of a phospholipid bilayer and also has other structures inside the cell enclosed in said membranes. The eukaryotic cell is much larger, and the most prominent feature of most, but not all, eukaryotic cells is the nucleus, which is where the DNA is stored enclosed in another membrane. Here's a sort of a diagram. One of the things we can really tell here is compare and contrast the size. The prokaryote is much smaller than the eukaryote. Here is that prominent feature, the DNA contained in the nucleus. And in this instance, the DNA is spread loose and diffusely throughout the entire nucleus. It takes up the entire volume of the nucleus and takes the blue stain. So this is the unwound state. What we have here is the cytoplasm, cell. Plasm is the liquid and other inclusions inside the cell. Here's the thin little membrane. And we see other structures inside the cell here. These little orange ones probably represent a mitochondrion. Hard to tell what these dark staining ones are. And the prokaryote has just its DNA just spread throughout the cell. Now, previously, I talked to you about viruses. And if there was a virus, see my little pointer here? The virus would be much smaller than even my pointer. Maybe the red part, the red circle. Maybe that little white dot inside the circle. That's how small viruses are. They don't contain much DNA, but they sure can reach, can't they? So the chromosomes are what we refer to as the genetic storage molecule. The chromosome consists of some DNA and proteins, but the proteins are just to help package the DNA. It's the DNA that actually carries the genetic information. Chromosome, what does that word mean? Literally translated, it means chromo is blue color. It's not chrome like the shiny stuff on a car bumper, old-time car bumper. It's the chromo means blue, and some is derived from the word somatic, which means body. So chromosome literally means colored bodies, blue bodies. Go back to that picture. This is a much different picture, but maybe along the way, I'll tell you what, if you want to see the picture I'm talking about, I don't have it right here. Google image cheek cell. A cheek cell, you'll see, looks like a plastic bag with a little blue round thing in the middle of it. And the little blue round thing is the DNA in the nucleus. And back in the 1850s, the first person to start experimenting with stains used one called methylene blue. And because of the microscopic level, there's not a lot of contrast. So we use stains to be able to visualize these structures under the microscope. And he used methylene blue, and he saw that this part of the cell stained blue. He had no idea what it was. And this is pretty. You don't know what it is or what it does. You find that later. First, you have to discover something before you can find out anything about it. So the name he gave it was chromatin. Literally, it means blue stuff. Blue stuff. And in the 1890s, some more people were studying cell division, and they saw these little blue-colored bodies, and so they kept with the naming tradition and named them chromosomes, blue stuff. So that's like literally where the name came from. In each chromosome is a series of genes. So in a typical human chromosome, there may be as many as 10,000 genes. A gene is the package or unit of inheritance. So this is what contains the instructions on how to make a protein. So you might have genes for a terrible example, but you might have genes for eye color. Of course you do. When we study genetics, we'll talk about the pea plant and how the pea plant has genes for whether it's going to be tall or short, or whether its pea pods are going to be green or yellow. And so those are discrete packages of DNA that code for a particular trait. The ability of cells to divide is the basis of all reproduction, growth, and repair of multicellular organisms. So reproduction, okay, so the cells we need to reproduce the cells. One cell makes two. As you grow, you make more actual tissue. So cell division creates more of the organism's body, and repair for multicellular organisms. Like for humans, our blood cells wear out. Our red blood cells have a lifespan of about 4 months. So that means they need to be, when they wear out, it's another story, but they wear out, we take them out of circulation. Mostly that's done by the spleen, but sometimes the liver. And as we take old, worn out ones out of circulation, we have to replace them. And and this is done in some special areas of the bone marrow where cells are very actively dividing to replace old worn out cells. If you get a scrape or a cut in the skin, cell division replaces that damaged material. So that's one important part about cell division there. Here is a false color of a cell undergoing cell division. And here we can see the DNA, these blue colored bodies, and this is in the chromosome form. So we find the DNA packaged into the chromosomes during cell divisions when we're going to move them around. Here's a little bit earlier in the process. Here's a little bit later when the cells get ready to pinch off right here. And we'll have a whole section on cell division later too. The DNA is in the chromosome state during cell division. It's in the chromatin state during the normal cell activities. So each chromosome is linear. That means it's one long chain of DNA, kind of like an extension cord. Long, thin, with discrete beginning and end. And many genes along the way. Like I said, maybe 10,000. And most genes encode information for making a protein. Other genes control when and where those proteins are made. I mentioned eye color genes. The only place with the eye color genes, every cell in your body, almost every cell in your body, has, almost all cells. See those red blood cells, they don't have anything, they get rid of it. So almost all cells have the gene for eye color, but it's not expressed in the genes of the skin of your thumb or in your liver. It's only expressed in the iris of the eye. So some genetic information is about when and where to express these proteins, make these proteins. All right, DNA is inherited by the offspring from their parents. Yes, that's where we get our genetic information, from our parents. And from the very beginning, the DNA controls everything. Cell division made, where it's made, development through the embryonic, fetal, child, everything, even when and where we might get disease. An interesting area of medicine these days. Now, I probably don't have to tell you this story. We've all been through it. Sperm fertilizes egg. Fertilized egg, called a zygote, undergoes cell division. Here we have the DNA in the nucleus. The only part of the, notice that there's less DNA in the egg. We get half of our DNA from our mom. We get half of our DNA from our dad. The only part of the sperm that actually enters the egg is the nucleus. The rest of this whole stuff stays outside the cell. The sperm is made of a head, a base, and a tail. And the tail of the sperm is called a flagellum. Acts like a little propeller, making the sperm swim. We'll talk about flagellum is singular. See, there's only one there. We'll talk about flagella, which is plural, a bit later in the cell part. And then it undergoes a series of divisions. And we go through the embryo, the fetus, and then childbirth. Oh, what a cute kid that is, huh? DNA molecule is made up of two long chains arranged in a double helix. So we say that DNA is a double-stranded molecule. That means it's got two chains linked together and in a double helix. So a helical shape is twisted like a spiral staircase. And so if we think of the DNA molecule as a ladder, think about it. You know, a ladder. You have the where you put your hands, and then your rungs where you put your feet. And so imagine half of the ladder is half of a rung and one of the rails, and then the other half is the other half of the rung and half of the rails. And then you take the ladder and you twist that ladder to a double spiral shape. That's the double helix. Along the rungs of the ladder of DNA are these building blocks of DNA called nucleotides, and we'll talk about nucleotides. We'll talk about DNA specifically a little bit later. And we'll talk about these nicknames A, G, C, and T, which are abbreviations for adenine, guanine, cytosine, and thymine. And we'll talk about each one of those when we get to the DNA section of macromolecules, the big molecules of life. So here would be half of the ladder. Here's one of the rungs, and here are the nitrogenous bases, these are called. The rungs are composed of a 5-carbon sugar and a phosphate. 5-carbon sugar represented by blue, phosphate represented by yellow, and these would be the nitrogen bases abbreviated. Now, at this point, don't worry about anything really regarding DNA structure. That comes at a later date. This is just a quick introduction. This is a double spiral shape where here are the rungs. So here is one of the letters, here's another, here's another, here's another, and we get the sugars and phosphates out here in a double spiral staircase shape. Genes control protein production indirectly. So the DNA stores the genetic material, but it stays in the nucleus, especially of eukaryotes, of course. And so all the material we need to build a protein, the raw materials, the amino acids, the ribosomes, which is the part of the cell where the proteins are made, the ATP molecules for energy, all that stuff is out in the cytoplasm of the cell, not inside the nucleus. And so while there's many genes along a chromosome, we pick, if we want one, we pick one and transcribe it into another nucleic acid. It's related to DNA. RNA stands for ribonucleic acid. DNA stands for deoxyribonucleic acid. They're very similar but very different molecules. And then that RNA goes out into the cytoplasm where that information is then translated into a protein. We have a whole section on that coming up too, so don't worry about this right now. This is just an overview where we're going. Expressing a gene, that's what I mean when I say that this gene is expressed, that means that this product is produced. We can think of the DNA. Now, back in the day, I used to talk about DNA being a cookbook, and that, you know, you open up the page and RNA is just one recipe. But nobody uses a cookbook anymore, do they? Hardly. Not even me. I get my inspiration, really. My favorite site is allrecipes.com. I can't make a recipe just the way it is. I always tinker with them, but I use them for inspiration. So there's a lot of recipes in the DNA that is allrecipes.com. When I find the one I'm interested in, I'll either put it on my iPad in the kitchen, or I usually just print it out because I'm old. So what was that? I just heard an echo. That was weird. All right, an organism's genome. We've had that word already. Its entire set of genetic instructions. The human genome has finally been sequenced, but mine is different from yours. And even so, my two friends that are in the living room waiting for me to pay attention to them, those would be my dogs. My dogs' names are Barky and Sean Lee. I know those are weird names. Their genes are different than their genes are as different from mine and yours, more different than from mine and yours and mine are. Ours are more similar. The genome between the dog and the human is much different. All right, yeah, I know. So you want to know where their names come from. Barky, my wife is enamored with Mark Wahlberg. Mark Wahlberg, you know, Marky Mark. And so she named the dog Barky Mark. just Barky for short. Anyway, he's my buddy. Me and him, we're like one. And the other dog, we're pretty close too. His name is Sean Lee. Yes, he is named after Dallas Cowboys number 50 linebacker Sean Lee. And the three of us are pals. So they're labs. Barky is a black lab. Sean Lee is a blonde colored lab, which is interesting. They're half brothers. They have the same mother. Maybe we'll get a picture of them some other day. Genomics and genetic study is usually done. The collection can be done out in the field or in a clinical setting, but then it also uses a lot of interesting lab machines with where you have to learn lots of sterile technique. And here's the girl working in the lab, and maybe if you want to live in a nice, clean, work in a nice, clean, air-conditioned area, something like this would be for you. If you leave Tarrant County College and transfer to another university or a university, maybe you can get a job working in the lab as an assistant. Usually when you're an undergraduate, and I'll explain what that means to you before you get a bachelor's degree, your ability to help is pretty limited. They'll let you, you have to work your way up. You probably start off washing stuff, but it's interesting to see the way a lab works. And I encourage you to, when you think of a field that you might want to work in, see if you can find somebody that works in that field, maybe even shadow them for a day after all the stuff is over, because you might find out that, yeah, I like to work in that area, or you might find out, no, I really don't like this area at all. And it'd be better to find out sooner rather than later, so it's never really too early to start. So let's stop this presentation right there. Pretty short one, and come back with the feedback. More information a bit later on the rest of this chapter. So we start off with aloha, hello in Hawaiian. Aloha is also goodbye in Hawaiian. So aloha